Hi everyone, welcome back to this session. So today in this session we are going to see about protoplast culture. In protoplast culture, the second step is culture of protoplast. So what is culture of protoplast? So in culture of protoplast, first step is development of a cell wall around the membrane of protoplast. Then the cell division which give rise to a small colonies. After that, the manipulation of nutrition and physiological condition, the cell colonies can be grown continuously as a culture. So there are different medium can be grown for culture. So here we are taking agar culture. Here the protoplast culture neither in semi-solid or in liquid medium. Agarose is a most frequently used agar to solidify the culture medium. The plating of protoplast is carried out by Bergman's cell plating technique, and in agar culture, the protoplast remain in a fixed position where it divide and form the cell colonies. Main advantages is clumping of protoplast is avoided. Liquid culture, this is one of the most preferred method and easy to dilute and transfer. The density of cells can be manipulated as desired, and the osmotic pressure of liquid medium can be altered as desired, and in some species the cell cannot be divided in agar, so in that condition we can use liquid medium. So next one is culture media. It is similar to that of culturing of plant cell like callus and suspension culture. Mostly MS and B5 media are preferred media, and special features of this media is the medium should be devoid of ammonium, quantity of zinc and iron should be less, and then the concentration of calcium should be 2 to 4 times higher than that of the cell culture, and it gives membrane stability. And when you speak about its hormones, there should be high auxin or kinetin which induces the cell division. Similarly, high kinetin which is an auxin which induces regeneration, and then glucose is preferred as a carbon sources, and there should be some amount of vitamins should also be added. Osmoticum and osmotic pressure. It refers to the reagents or chemicals that are added to increase the osmotic pressure of a liquid. Isolation and culture of protoplast require osmotic protection until they develop into the strong cell wall. Freshly isolated protoplast added to a medium, it may burst, so addition of osmoticum. Types of osmotica. First one is non-ionic osmotica, which is a soluble carbohydrate like mannitol, sorbitol, glucose, fructose, galactose, and sucrose. So in the following carbohydrate, mannitol is a frequently used. And second is ionic osmotica, potassium chloride, calcium chloride, and magnesium phosphate. Culture method. So same as that of cell culture, with little modification we can go for feed layer technique, which is used for selection of specific mutants and then exposure of culture to X-rays and plating on an agar plate. So second one is co-culturing of protoplast. Here the protoplast of two different plant species. One is slow grow process, another one is fast grow. So two different plant species are cultured in case of co-culture of protoplast. And third one is microtroph culture. It is a specially designed Kuprak device is used here. The inner chamber, which has several wells, nutrient medium can be added, and in outer chamber which will be filled with water to maintain the immunity. And next is regeneration of protoplast, which is regarded as a protoplast development occur in two stages. One is formation of cell wall, another one is development of callus or old plant. That is, formation of a cell wall is a process. Start with few hours of isolation and which takes nearly 2 to several days. When a cell wall develops, it loses its spherical shape, and newly developed can be identified by chalcophore white fluorescent stain, and newly developed cell wall contain loosely bounded microfibrils, and there should be continuous supply of nutrients is required. Only then the cell wall will be formed. Development of callus or whole plant. So this is the next process. When cell wall formation around the protoplast is complete, then the cell will be increased in size. First division occurs within 2 to 7 days. First the small colonies, then at the third week large colonies will be formed. This colony is then transferred to a osmotica free media, which is mannitol or sorbitol free, for further development to form callus. With induction and appropriate manipulation, cells can undergo organogenic or embryogenic differentiation to form whole plant. So this is how the regeneration of protoplast will be takes place. So first we will be isolating, next we will be culturing, at last it will be regenerated into the whole plant. Thank you.